Hi, and welcome to our video on cellular respiration. In a previous video, we looked at photosynthesis. If you haven't watched that one already, you're probably wise to go back and take a look at it before you dive into this one, or maybe you could do them in reverse. I don't know. Your mileage may vary, but let's get into it. In this video, we're going to talk about the following: how the process of cellular respiration converts the energy that's stored in macromolecules into ATP, which we know cells can use to power cellular work. More on that in a previous video as well. We're going to discuss the matter and energy transformations that occur during the major stages of both types of respiration, and then we're going to look at the role that respiration plays in living things. This is the overall summary equation for aerobic cellular respiration, and what I've done here is I've color-coded the atoms so we can see where they start and where they wind up. This is showing us the respiration of a molecule of glucose, and we're going to use oxygen during the process. And that's going to convert the glucose and the oxygen into water and carbon dioxide. You have some intuitive understanding of this because you eat food and you breathe, and you know that you breathe in oxygen and you breathe out water and carbon dioxide. But I'll also point out that this equation is really only the summary equation for aerobic cellular respiration. And there are actually two major versions of respiration that happen in cells. There's anaerobic respiration, which does not use oxygen. And then there is aerobic respiration. We are aerobic respirers as animals. All animals are aerobic respirers. But most cells on the planet actually just carry out anaerobic respiration. Most bacteria respire anaerobically. Regardless of how it happens, it always has this kind of a structure. In anaerobic processes, we start with glycolysis, and then we undergo a process known as fermentation, which enables us to continue glycolysis. This is a self-contained cycle, and this cycle will produce about 2 ATP molecules for every time that you spin that cycle. But any organisms on the planet that want to carry out aerobic cellular respiration, in particular multicellular plants and animals, are also going to start with glycolysis. But after glycolysis, they're going to go into two other steps called the citric acid cycle, and then the process known as oxidative phosphorylation before being able to go back to glycolysis. So either way we look at this, we have cycles, but the difference here is that whereas anaerobic respiration carries out glycolysis and then fermentation and generates 2 ATP, in aerobic processes we're going to start with glycolysis also, and then go to the citric acid cycle, and then oxidative phosphorylation, and that's going to make about 20 times as much ATP. The number is not specific. We don't actually have a specific number, but we usually say it's anywhere from 32 to 38 ATP molecules, which is roughly 20 times more that you get using aerobic respiration. Of course, no matter which process we're talking about, they all start with glycolysis. Glycolysis is one of the oldest metabolic reactions in the living world, and it happens in the cytoplasm of every cell on the planet. It's actually a very complicated process. There are many steps, and each one has a specific enzyme that takes one molecule and converts it into one other molecule. We're not going to focus on that. I'm not going to show you the diagram just because I think it will confuse you. But what's going to happen in glycolysis, if we're starting with a glucose molecule, is that glucose is going to be converted into 2 molecules of pyruvate. If we think about glucose, glucose has 6 carbons, and then we're going to get 2 molecules of pyruvate. So no magic here, right? We're going to have 2 3-carbon molecules as a result. And this is also going to produce 2 ATP. We actually produce 4 ATP, but we use 2 to begin. So we actually say that we produce 2 net ATP. So tracking our inputs and our outputs, we're going to put in a molecule of glucose, and we're going to get out 2 molecules of pyruvate. We're going to put in 2 ATP, and we're going to get out 4 ATP. And we're also going to use some electron shuttles here. We're going to take some electrons off of glucose and store them in 2 molecules of what are called NADH, which are electron shuttles. And this is good enough for almost every cell on the planet. You're getting more ATP out than you're putting in, and so you can run cellular work if you don't have a particularly high energetic need. Of course, multicellular organisms do have that, and that's why we're going to do something else. But for almost every cell on the planet, glycolysis is fine. It works to provide enough energy so that the cells can do the things that keep them alive. The problem, if you are anaerobically respiring, is in these NADH molecules. There's no way to get these NADH molecules at the end back to what they started with, NAD+, unless you go through another process. And that process is known as fermentation. So what's going to happen in fermentation is that the NADHs are converted back into NAD pluses. And in that process, we're going to take our pyruvates and we're going to convert them into other molecules. And in that process, we're going to take electrons from the NADHs and put them into our pyruvates. And our pyruvates will then be converted into some sort of end-stage molecule. Different organisms on the planet do different things, and they convert their pyruvates into different end products. I'll just focus on two of them for the purpose of our discussion, but there's like hundreds of different possible end products that different organisms convert their pyruvates into. Yeast cells, which are a type of fungus, convert their pyruvates into carbon dioxide, CO2, and ethanol, which is alcohol that people can consume. It's widely used, and the carbon dioxide that yeast produces during this process is used to do things like allow bread to rise, etc. So this is a very important commercial reaction that takes advantage of fermentation. And actually our muscle cells, so animal muscle cells, can temporarily carry out anaerobic respiration if they need to, if they're not getting enough oxygen because you're running very fast or whatever it is, and they're going to convert their pyruvates into lactic acid. If you do this too long, the lactic acid builds up and your muscles will cramp, but as a temporary fix for not getting enough oxygen, that definitely can happen, though only in your muscle cells, not in the rest of your body. Don't hold your breath for too long. You'll find out what happens. So our inputs into fermentation are going to be our pyruvate. We'll have two of them, and we have our two NADHs, and our outputs are going to be two end products of various compositions. We see a couple up above, and we're going to have our two NAD pluses. Notice that we don't make any ATP. The only purpose of fermentation is to regenerate these NAD pluses so that cells that are anaerobically respiring can continue to produce ATP through glycolysis. Let's pause for a second before we go into the aerobic route, and let's talk about where aerobic respiration occurs. In eukaryotes, the site of aerobic cellular respiration is the mitochondrion. We have a diagram of a mitochondrion here. We're going to focus on two different regions. The first is this space inside of this inner membrane. It's what's called the mitochondrial matrix, and it is where the next step in the process, the citric acid cycle, occurs. The other region that we should really pay attention to is this inner membrane, because that's going to be the site of the last step, which is oxidative phosphorylation. Again, we could get really, really complicated with this stuff, and it's much more complicated the more in-depth that we get, but I don't think we need to focus too in-depth on it for the purpose of this video. Happy to link you to other resources if you need them, if you want to know more about these processes. The other point that I'll make is that many bacterial cells can carry out aerobic cellular respiration, and they clearly don't have internal mitochondria. They carry it out at their cell membranes. And just like in photosynthesis, where we have evidence that chloroplasts were once bacterial cells that have since evolved into modern-day chloroplasts, similarly, we also have evidence that strongly points to an independent evolution of mitochondria from another group of bacterial ancestors, which is pretty cool if you think about it, because every cell in your body has multiple mitochondria, and they're all the descendants of these once free-living bacterial cells, all living inside of our eukaryotic cells. But of course, for the purpose of this video, we are going to focus on the citric acid cycle in eukaryotes, and so that's going to happen at the mitochondrial matrix. So if we think about where we left off at the end of glycolysis. We're not going to go through fermentation, and so we're going to start with pyruvate. And actually, before pyruvate enters into the citric acid cycle, it's going to lose a CO2, and it's going to become an acetyl group. So pyruvate had 3 carbons. We've removed 1 for CO2, and so now we have a 2-carbon acetyl group. That acetyl group is actually what's going to enter into the citric acid cycle, and over the course of the citric acid cycle, that acetyl group is going to be turned into 2 additional CO2 molecules. When pyruvate became an acetyl group, we also produced an NADH there, so we have another one of these electron shuttles. And then during the citric acid cycle, we're going to produce another 3 NADHs. We're going to produce another electron shuttle called an FADH2, and we're also going to produce an ATP. So if we think about our inputs, we had pyruvate being put in, but pyruvate, of course, was first converted into an acetyl group, and it released a CO2 molecule. Our output is going to be an additional 2 CO2 molecules from that. We got 1 NADH in that pyruvate conversion. And during the citric acid cycle, we're going to get another 3 NADHs and 1 FADH2. and we also got ourselves an ATP. But you're probably doing the math, and you can understand that we made 2 in glycolysis, and here we made 1 per pyruvate. Of course, if we were going to do everything from the original glucose, we would need to multiply everything by 2, so we'd get 2 ATP in that case. That doesn't sound like 20 times as much ATP because it's not. We're going to get most of that ATP in the final step, oxidative phosphorylation at the inner mitochondrial membrane. What's going to happen in oxidative phosphorylation is that all the electron shuttles that we produced are going to release their electrons, and those electrons are going to be used to produce ATP. So electron shuttles release electrons, and those electrons are used to produce ATP. The other thing that's going to happen with those electrons is that after they're used to produce ATP, they're going to be combined with oxygen and become water. So we'll have to add some protons to it, just so I'm not accused of any sort of chicanery here. So if we think about the number of NADHs that we're going to put into this process, in glycolysis, we made 2. So everything from that point onward, we have to double. So we made 2, converting them into acetyl groups, and then we made 6 in the citric acid cycle, again for 2 pyruvates. So we have a total of 10 NADHs that we're going to put into this process. We also made 2 FADH2s in the citric acid cycle, and we're also going to need oxygen gas in this process. So we're going to put in these 10 NADHs, these 2 FADH2s, and as much oxygen as we need. And what are we going to get out? We're going to get out ATP. Roughly, we get 3 per NADH and 2 for every FADH2. So if you think about that, we've got 10 NADHs, so that's 30, and 2 FADH2s, so that takes us to 34, right? So about 34 ATPs produced. The reason why it's about is because they're not directly connected to each other. We've converted our NADHs back into NAD pluses. We've converted our FADH2s back into their form so they can go back and get more electrons, FAD. And of course, we convert the oxygen into water. You probably notice that the difference between something like NADH or FADH2 and then the form it becomes are those protons, those H's. And those H's are going to be used in this process, and they will combine with water at the end. So at the end of oxidative phosphorylation alone, we have 34 ATP. We made another 2 during the citric acid cycle, and we made 2 that we netted during glycolysis. So that's about 38 ATP, which is roughly 20 times the amount that we would have gotten had we just stayed with anaerobic cellular respiration. And that's why multicellular organisms need to carry out this process, because multicellular organisms need to coordinate the action of hundreds of trillions of cells in cases like you or me. And in order to do that, we're going to need a lot more energy than we would if we were just a single bacterial cell. This ATP that we're going to make is then going to be used to power cellular work. We've talked about that before, but what is cellular work? It's everything that cells need to do in order to remain alive. And it's a crazy, crazy thing how much ATP you make over the course of a day. The estimate is that approximately you make your body mass in ATP over the course of the day. That ATP is, of course, immediately used for cellular work and converted back into ADP, so it cycles around. And it's constant, so you don't gain in mass as a result. But if you think about that, every day as you do your thing and remain alive, you are producing an equivalent amount of ATP to your body mass. Pretty amazing. As we wrap up this video on cellular respiration, let's pause here and make sure that we can do the following things. Make sure that you can explain what happens during cellular respiration, including what happens in anaerobic respiration and aerobic respiration. Make sure that you understand where all major steps of both processes occur in eukaryotic cells, and the inputs and outputs of the major steps. Make sure that you can describe how each substance in the summary equation for aerobic respiration is used or consumed in the process, and also take a moment and compare and contrast anaerobic respiration and aerobic respiration. How are they similar? How are they different? That kind of thing. If you can do all that, you're doing great. If not, that's okay too. Take a moment, pause the video, and write down any questions you have so you can get the answers that you need. Thanks again for watching. I really appreciate it. Have a good day.